a short overview of dementia, cognitive impairment, and mainly cognitive rehabilitation in the context of a multimodal management of dementia. What is dementia? It is a symptom and not a diagnosis, and it occurs in most cases during the process of aging. We have more than 100 types of dementia syndromes. However, the most common is Alzheimer's disease. And as you can see, it is an umbrella symptom, and we always need to keep an eye on vascular or systematic diseases that may have the same outcome when investigating Alzheimer's. Why is it important? Because it has a huge epidemiological stamp. We have almost 50 million people globally suffering from this, and there is also a very high cost, almost $820 billion just a few years ago. And as we mentioned, it is an umbrella term. We have different types of dementia, and in most cases, the diagnosis is clinical, and it has to do with the presentation of the patient and what his relatives or carers mention. We have a lot of tests that help us quantify this. However, none of them is as sensitive and as specific at the same time we would like it to be. For example, we have the mini mental state examination. This is very sensitive, and it's very good to classify people in mild cognitive impairment and Alzheimer's, but it's not so specific. We have mini cog test, we have Montreal cognitive assessment, and many other modalities. And a very important entity, which is actually an alarming sign for Alzheimer, which is the precursor of dementia, is mild cognitive impairment. And this is the common frequent thing that old people tend to forget. If we quantify this in a mini mental state exam, we can find that about 10 to 20 people over 65 may have mild cognitive impairment, which leads with a high likelihood to Alzheimer's. When it comes to the treatment, this is another problem because we have pharmacological and non-pharmacological, which are psychological and behavioral treatments. But in both cases, we have low efficacy, and they focus on disease stabilization rather than regression. So this is a huge deficit. And for this reason, in the last years, we have seen several training approaches to come up trying to fill this gap. And most of them are missing evidence because they are approaches that one university or one practitioner developed, but they have not been validated in the context of randomized studies. And this is why we are trying to investigate today an aspect of multimodal treatment, which is cognitive rehabilitation. So we are trying to investigate the evidence about cognitive rehabilitation, which is a very interesting approach to Alzheimer. And we have done a literature search in PubMed. You can see our keywords and methodology. And without further ado, I will introduce you to cognitive rehabilitation, which is a comprehensive approach combining cognitive stimulation and cognitive training. Its main benefit and advantage is that it focuses on assessed behavioral and social disability in total, rather than specific deficit like I'm forgetting names or I can't recall numbers and so on. It is also an umbrella term as well, an umbrella term for an umbrella condition, which includes a lot of strategies and specific interventions that can be personalized depending on the case and on the experience of the therapist. And when it comes to its scientific validation, we have three kinds of studies. Cognitive rehabilitation without any memory aid, cognitive rehabilitation with non-electronical external memory aids like a piece of paper or tools and so on, and studies using technology. And we have a representative sample, probably the most recent and accurate studies, or the biggest studies, to be honest, not the most recent. When it comes to only CR sessions, we have an observational study by Zahmen and colleagues with 52 people who were assessed for 1 year. And even only sessions without any aidings showed that slight global cognitive decline, which means that there was a decline, but it was slighter than it is usually in most people with this condition, and also lower level of dependence, which is very important for the care of these people. When they added practical components, and this happened to a study of Clara et al., we saw that in a randomized control trial, which is very good for evidence, item-assisted cognitive rehabilitation was effective against early-stage AD. And this led to disease stabilization, which adds to the quality of life of the people in the next years. And when they tried to add technology, also in an RCT, they found out that long-term results could have been achieved because there was long-term evaluation of these patients. However, there was the bias that people were aware of the treatment modality they were getting, and this makes them more proactive or motivated. In any case, and in all cases, every coin has two sides. We have benefits and drawbacks. Cognitive rehabilitation is non-interventional, non-pharmaceutical treatment. This means no side effects. It is personalized, and it is highly accessible, both to a wide spectrum of specialties, medical and non-medical, allied health professionals like ergotherapists, psychologists, and so on, who can be trained on it and offer it to patients all over the globe. However, its efficacy is therapist dependent. Not everybody will have the same experience and the same training. And it also only slows down the progression of the disease and does not regress it. Probably in combination with other treatments, it may help to regress this condition. What is the take-home message from this? In the future, we will have to investigate further in a global scale the direct and indirect cost, because everything today is cost effectiveness, and we won't have this treatment going out if it's not cost effective. And this stated it needs to be acknowledged by insurance agencies, state or private ones. It is very important that we can use both everyday materials and sophisticated computer software. This is a huge opportunity for research, but also it's a great opportunity to train people in their everyday life to use things in order to enhance their own memory. And in the future, there are a lot of prospective studies about errorless training, making this treatment more specific and not tolerating mistakes in recalling processes, which seems to be quite encouraging. And I don't know, did I forget something here? Yes, I forgot to thank you all for your attention and for the floor. We are open to your questions.
Thank you very much, Christos.
I will also mention that we have written a chapter in a book published by Springer about this. I will share the link right after in the chat.
Please do, because it's extremely interesting, and I'm sure I'm not the only one that needs to do a bit more of reading on the topic. And if I may ask a question while we wait for to start flowing in, since you have surveyed the literature, you know more than anyone else right now about cognitive rehabilitation in AD. Do you have a sense of what percentage of those studies were focusing on memory, and how many were actually targeting also other cognitive function?
Well, yes, the majority of them included memory. So I can say that more than 90% of the studies that we read were about memory, but most of them also investigated other parameters, either cognitive or behavioral. For example, the interaction with the carers, which was the key topic in the first study, and how the dependence was lowered in the course of the treatment. And there are also some confounders in these studies, especially when it comes to the use of technology. Elder people who suffer from this condition have a low level of digital literacy in most cases. So whenever memory, we don't know whether it was a problem of memory, a lesion of memory, or also a problematic interaction with technology and computer interface.
Right, that's very difficult to tease apart, right? That's a big confound.
Indeed.
And talking of technology, of course, the idea that things can be computerized and done online, so you can administer the therapy or the task anywhere in the world, that's a huge plus. But were you thinking also, when you say that it's important to integrate technology, are you also thinking of like neurostimulation and this kind of technology, or you're mostly talking of computerized tasks?
Well, absolutely, because cognitive rehabilitation is read and investigated in the context of a multimodal approach, a pharmacological and non-pharmacological approach. So any kind of technology is welcome. When it comes to solely the cognitive rehabilitation, to be honest, they focus on aidings that come from technology, for example, quizzes in the computer, or some specific games that help memory. And to be honest, when we were writing this, we didn't know about the pandemic. So right now, I believe that computer interface and technologies like Zoom, WebEx, and so on will play a role in this as well.
Absolutely, it's become more and more important. Yeah, that's very true. I just remembered that I forgot something, my bad, which is that people can type their questions or they can raise their hands, and we can even bring them on screen if they prefer. And the same, the same for the speakers if anyone wants to jump in, because I don't want to monopolize the floor. I am very curious because I work with dementia, not typical Alzheimer's. I work with atypical dementia, like frontotemporal dementia and primary progressive aphasia. So I know a little bit about the therapy that are proposed in that field, but not specifically for the standard, the typical AD, and so that's why I was asking about memory. And now other cognitive functions can be used to like compensate maybe the memory loss.
Yes, yes, of course. And to be honest, there is always a problem with the tests because you can really draw a red line between memory, cognition, and behavior.
Absolutely, absolutely. And I'm guessing in your meta-analysis, since it was done in 3 languages, right? Like you were searching in English, also
Yeah, of
French,
course,
I
most
remember.
of the studies were in English, but because we also speak Greek and French and German, we had the chance to come to read some studies published only in these languages.
That's very interesting. Thank you very much. This was a very, very insightful. Please stay connected so you can ask questions to
Of
our
course,
next
yes.
speaker. Well, thank you very much.
Thank you once more.
Also, keep an eye on the Q&A because people might still come up with questions and you can type down your answer
Yes,
there.
of course, and I will share the link of the book right now as well.
Exactly. Please do. Thank you very much.
Thank you.
We are doing great on time, and we can switch to our second and final speaker for this session. Professor Hugo Spears is joining us from UCL Institute of Behavioral Neuroscience, and he will be teaching us about the neural and cognitive and cultural foundation on navigation ability, something I know very little of, so I'm very curious, as most of you, I'm sure. So Hugo, if you want to start sharing your screen, we can jump right into it.
Great. Okay.
That looks great. Thank you very much.
You can see my screen. Great. Oh, thank you so much for organizing this. It's a fantastic conference. So yeah, as was mentioned, I'm going to really ask the question of what determines navigation ability? Why are some of these people in this group potentially better than others at navigating? Is it their brain structure? Is it the neural coding they have? Is their age and gender important? And indeed, is the culture that they've grown up in or exist in affecting how well they can navigate? In terms of brain structure, a key study 20 years ago is the discovery that London taxi drivers, here's a photograph of one, actually have a larger posterior hippocampus. And this is interesting because they spend 4 years memorizing 58,000 streets that they have to navigate without using digital aids to use their license. And that's been replicated. We compared bus drivers to taxi drivers and found, bus drivers are boring, taxi drivers are very large hippocampus. And indeed, Ellen McGuire has shown that their hippocampus changes longitudinally with the training they have. But for the rest of us, the mere mortals amongst us like me, there's not a huge, some studies have shown some relationship between brain structure and navigation ability, but others more recently have suggested also that it's not clear that there's a link for healthy younger individuals. So there's a bit of a mixed picture currently between brain structure and navigation ability, and it's something we need to investigate. And indeed, my group are currently actively studying taxi drivers and non-taxi drivers to make that leap. But what about neural coding? Why might one of your friends be so good at navigating and another one not very good? In a study we ran 5 years ago, we had people navigate around a very simple environment, a courtyard, and they had to think about different directions to goals, and inside that, we could decode which way they were facing and where their goal was from the entorhinal activity using a representational similarity analysis of the fMRI signal. But what's important here for the story I'm telling is that the precision and the reliability of that direction signal in the entorhinal region could predict to some degree how accurate our participants were. It's a very small sample, but it mirrors evidence from Christian Dohler and colleagues showing that the grid coherence signal they could detect in their fMRI analysis, the stronger someone's directional coherence in their entorhinal cortex, the better their spatial memory. So this is very preliminary evidence really over a few studies, but it suggests that maybe the stability of the neural code for direction might be one of the reasons why some people are better navigators. But I'm going to spend the rest of the talk now looking at the sort of more the questions about differences between people in terms of demographics and the culture, as to why those might play a role. And in doing so, I'm going to introduce a study we started 4 years ago. In 2016, we released See Request, an app for mobile phones and tablet devices that anyone could download from the App Store and Google Play. And it was in 17 different languages, so it really was aiming to spread wide. And one of the main reasons we developed this was again from the previous talk by Christos, that it'd be very useful to have some monitoring tools for spatial navigation problems in Alzheimer's dementia. It's a key early symptom, and the aim of this was to try and build something that would be useful. So in the game, the main part of it, you're playing a little boat and you're trying to navigate it around through a virtual environment to get to checkpoints. And here's two schematic examples of environments that we put down, like top bird's eye views. But I'll now show you level 17 of it. We have 75 of these levels in the game. And here's two people playing these different levels, and you can see they're passing through checkpoints. There's various landmarks, but what key thing here is that we're tracking their position in the game, and we can tell how accurate every moment is in terms of their navigation. We can score everyone that plays this game on their ability to navigate through these environments. Fortunately for us, it was funded by Deutsche Telekom, T-Mobile. We could reach out and get many people, and we have 3.9 million people's data now playing this game. And the person I'm going to introduce here, Antoine Coutreau, who's a French scientist who was the postdoc who began the work on this and now runs his lab looking at various other things, including this, has really been going through this data in great detail. He published 3 papers in the last few years, and we have a preprint I'm going to spend time talking about today. But really, for the purposes of this, you can downsample 3.9 million people substantially to just 150,000 to kind of reveal the pattern in the heat map of a given level. But if we downsample that to just 9 people, you can see someone in the bottom right here is absolutely appalling. They're going all around, just never really getting to the targets. Whereas the person at the top here is just phenomenally good. I just know exactly where to go without error. Why? What could explain these patterns of variation? The biggest explanatory variable, unfortunately for many of us, is age. There's just a linear decline across the world population in terms of age from the earliest stage. It's not like you have to wait till later in life, it just declines. We see a consistent male advantage across participants around the world. When we fit linear models to try and explain why might populations in some countries be navigating better than others, we start to reveal some explanatory features. So what we found, and we published 2 years ago, was that actually on the y-axis here is the conditional modes rank from a linear model. So this is just in a rankings table from a very carefully done analysis accounting for other demographic features. So you can adjust for features like age and gender. How well different countries do. So Finland tops the league for the best navigators in the world. And we were trying to explain why might they be doing so well. And it seems like the gross domestic product of a country has a strong, powerful feature into explaining why some groups of people might navigate better than others. More wealth, more healthcare, all things that are associated with GDP seem to play into giving people better navigation skill. So hopefully this might change more equality around the world in terms of access to these things. We should also note that this isn't linked to just raw video game skill. You don't get this correlation if you're just good at video games. This is specifically to do with navigation ability. And so The other key thing we wanted to know is, why are we seeing this male advantage? And the male advantage, the male difference between men and women is huge in some countries and tiny in others. Why? Why is there a male advantage more in some countries? What we found quite strikingly is, if you take the World Economic Health Forum's gender gap index of how unequal each country is, you can predict to some degree the rankings for why the difference between men and women. So Norway and Finland, there's almost no difference between men and women in their navigation ability, and they have the least gender inequality in the country. So hopefully this chart may change if we can move to more equal societies. This is a strong driver for any studies looking at sex differences and things in cognitive behavior, is that it may well depend on the country you're growing up in, the culture that people are raised in terms of what we might impact on that. And we also should note on route through this talk that we've also looked, Gillian Coughlin in Michael Hornberger's group. Michael's the co-lead on SIA Request, and his team study dementia in the University of East Anglia has shown clearly that there's a negative impact of genetic risk for Alzheimer's. So our particular wayfinding measure is sensitive to, so these are people who are perfectly healthy, but they are at significant risk of developing Alzheimer's. And indeed, they hug the edges of environments as they navigate more than people who are not at risk. But we hope this might turn out with more research in the future to tell us more about aiding Alzheimer's research. The last question I want to address in this short talk today is, how does the environment you grow up in impact your navigation ability? We've seen how GDP and gender inequality can, but what about if you grew up in a city or in a rural environment? There are big differences in what you experience growing up. So is there an advantage to one of these? And we found there is a difference. So what I'm going to show now is on the y-axis in a moment is navigation ability. So high on this axis is good navigators and low is bad. And I'm going to plot that over age. So you'll see the horrible dramatic loss with performance with age. We'll show you the male advantage where men are doing better, but I'll also now separate this so we can see dramatic loss with age and the male advantage that men are doing better. But the green line and the gray line show you the advantage you have of growing up outside a city if you're navigating. So it's not as big as the gender divide, but what we can see is that a woman who's grown up outside a city in her 70s is equivalent to a 60-year-old who grew up inside a city. So there's a real genuine difference here in terms of the impact of this. But not all countries show this strong effect. This is the world average. And so some countries have a very big effect and others a much smaller effect. For example, there's not much effect in Greece, where Christos is, I imagine, sitting. But there is in the USA and in Argentina, there's a very strong effect. Why? Well, we realized that cities vary a fair bit around the world in one key feature. There are many features, but one key analytical feature you can see is how gritty they are. So Chicago in the bottom right here, its streets are highly organized. They're really aligned northwest, east, south, and you can measure with Shannon's entropy how disorganized or organized cities are. So what we did, I'm going to show you on the y-axis of the graph, is countries of the world plotted by their street network entropy of the top 10 cities, extracted by Antoine going through OpenStreetMap. On the y-axis, the negative impact of growing up in cities. And you can see there's a negative correlation between these two variables. So growing up in a city with high street network entropy means less negative impact of cities on your ability. So people growing up in Argentina are having a worse impact of their street networks on their navigation, it seems. So it seems that might be, that gives us evidence that it's the structure of the environment that may be playing a role. We decided to go a bit deeper than that. And in my last slide, I'm now going to show, we'd look back at the game levels. So some of the levels you saw in my movie earlier are quite city-like in their structure. Others are much more convoluted and have a high entropy. And so what we can do is plot those along this x-axis here of how the different points on this graph relate to our performance in different game levels against the negative impact of cities. So what we see is that, yeah, as we'd expected, growing up in cities generally has a negative impact on navigation ability in our complex levels. But actually, when you go down to more simple, more grid-like levels, there's even an advantage potentially of having grown up, say, in New York City or Chicago on your ability to find your way. So it's not all a bad thing growing up in gritty cities. Our data overall, the take-home message in this talk is really that humans appear to adapt their navigation ability depending on their environment. And we're looking at this in lots of nation states, 38 countries across different languages and cultures, and large samples. I've also talked at the beginning of this talk about the need to understand greater the link between our neural representations and brain structure and navigation ability. We're beginning to see effects. And I mentioned there's a clear impact of age and indeed gender, but the gender impact is strongly linked to inequality. It's not, it's almost negligible when you account for that. Finally, I'd just like to acknowledge all the people that provided key input to this research. I'm talking about particularly Antoine Coutreau, who did all the painstaking work analyzing the data with us, and Michael Hornberger, who's my key lead on SeaRequest, and Ed Manley, who's the geospatial analyst who's put a lot of time into the work, and also the funders who've made it all possible to actually do this work. So I'm happy to take questions now from anyone listening. Thank you.
Thank you very much. Amazing talk and great wealth of data. I see already questions coming in, so I'm gonna have to keep mine for afterwards. Alan Andred, sorry, he's asking, what about educational level? What do you suppose it has an impact?
That's a fantastic question. So I forgot to mention as I went through that yes, we modeled, I didn't show on the graphs, we also model in their education level, because you might worry that people who grew up in cities have less education. And indeed, we were able to account for that as a covariant. We find that we still see a negative relationship with the entropy, even when we account for education. So it's not purely driven by education. Education has a pretty similar level to that home environment effect size on wayfinding. So it certainly does. We knew it's a key variable if you're going to be looking at diagnostics for something like dementia. Yeah, and it's something we're indeed writing up with some very interesting analysis you can do with education. So you can look at where countries raised, they changed the education level. So in England, it changed 1 year. Right. And that we can see has interesting effects going back through the data, but we'll wait to submit that for preprint in the next years. I can see other questions coming in.
Yes, exactly. Avital is asking, thanks for the great talk, first of all, and wondering if you have any access to subject attention levels.
Oh, that's
this kind of psychological trait may impact the navigation abilities.
Oh, that would be lovely. So we don't have that. That's exactly the kind of thing where, having now done this large scale study, we hope to release a version of this application for other researchers to use alongside ourselves, where we can ask lots of questions very carefully about traits, levels of things like anxieties, and other questions we're interested to look at.
Absolutely.
We don't currently have that, but it's a really good question. But we don't have a measure in our current game for attention levels. And indeed, you can see people, if you look at performance, some people just sort of wandering off. And I would love to look at what's happening in those participants and who are they, and can you predict that? But we haven't made it that far yet with the data. It's with 3.9 million quite large.
Yeah. Alan has a follow-up question, is wondering whether the difference in living in the countryside until 5 years old or 10 years old, like when should you move out of the countryside?
Right, so that's a great, we don't know the answer to these questions, but these are the kind of questions that will be extremely well answered, I think, through using birth cohorts, where you have the postcode that somebody lived in from their birth to the point you test them, to see if you can model that. But we don't know yet whether the general upshot of it is, it looks like if someone says they generally grew up inside a city, it overall has a negative impact on most complex environments. But if you get a bit of other things, like you grew up outside cities in the suburb, then it's better. It's people are performing better.
Yeah, I was trying to do the math in my head. I'm a female, but I grew up in the countryside.
Yeah, exactly. We can pinpoint you. We'll have thousands of people who we can compare you to. So you have more questions, yeah.
Exactly. Ameya Menon is asking, for the video game, did you see any improvement in navigation ability over time over varying maps?
Oh, great. So we have looked at that. After this meeting, I'm going to have a meeting to discuss how we can move forward to pre-printing a paper looking at that, where, if I remember correctly, we see people who are not very good navigators benefiting more from repeated experience in it, which is nice to see. It's not just everybody benefits from retrying. A nice feature of the game was that there was, you really had to work hard to get 3 stars. So people repeatedly played levels, and we'll be able to take apart that in a lot more detail as we move forward. I should say as well, raises the point, if anybody is interested, they can get in touch with me to potentially use the app, a research version of it, for a study they want to run, or to analyze the massive data, which we hope to make available very soon.
Exactly. This is actually the last question that just came in from Denise Enriquez, and it's also my question. What can we do as researchers to get our hands on the app?
Yeah, just literally contact me through Twitter or email. If you just Google me, you'll find me available. We'll be delighted to help other teams. We are collaborating with lots of different people with See Your Request, but we'd be happy to collaborate with more. And the large 3.9 million data set that will be made available soon once we get that recent preprint published. That's the aim.
That is amazing. I'm sure you will be flooded with messages and requests now, and we will all keep an eye on that preprint. I believe I did share the link, but yes, I did. But yeah, we will all keep our eyes on you. I want to thank you very much, and Christos as well, if it's still around, for the two beautiful talks. I learned a lot.
Thank you very much, and thank you very much, Professor Spears, for this talk. It was amazing.
Likewise, I enjoyed yours, Christos. Very, very nice. Yeah, thank you.
Thank you very much. Also, thanks to our attendees. And now you all get like 10 minutes of break before the next session of Neuromatch. Thank you, everyone. Bye.
Well, it's a little dark for me. God, this is terrible. Okay, I think we should get started. So our next speaker is Kaylin Chadwick. And just a second. She's coming to us from the University of Alberta, Canada. She's going to be speaking about spinal-mediated kappa opioid receptors. Antinociception is blunted in a mouse model of multiple sclerosis. So thank you, Kaylin. And if you can go ahead and share your screen.
Wonderful. Thank you, Alex. All right, can everyone see my screen? Perfect. So, as was just stated, my name is Kaylin Chadwick. I am a master's student in neuroscience at the University of Alberta. I'm co-supervised by Drs. Anna Taylor and Bradley Kerr, and my research is looking at the kappa opioid system in a mouse model of MS. So what is MS? There we go. It is a disease of the central nervous system that is characterized by inflammation, autoimmunity, and demyelination. And importantly for my project, about 60% of people living with MS experience pain as one of their symptoms. So how does our body control pain? Well, that would be the opioid system. Now, when I say opioids, the first thing that pops into your mind is probably the opioid crisis that we're currently living in. However, this is due to agonists of the mu opioid system. So morphine, fentanyl, heroin, those all act on the mu opioid receptor. However, currently there are studies looking at the use of the kappa opioid system as a potentially less addictive counterpart. So we know that agonists of the kappa opioid receptor, or KOR, are analgesic. However, our lab is concerned that multiple sclerosis may cause a downregulation of KOR, which would be a problem if we're going to use it as an analgesic. So the reason that we think this might be the case is that in 2008, a study was published showing a decrease in kappa opioid receptor expression in the spinal cord in EAE. Sorry, not in EAE, in a model of MS. So we wanted to see whether KOR agonists are still analgesic in MS, even though the study showed a decrease in the receptors. So to do this, we used EAE, an experimental autoimmune encephalomyelitis, which is slightly different than the one that the previous study showed. However, we wanted to use it because we know that it shows the hallmarks of MS, as well as pain, which is kind of important if we're going to study analgesia. So the first thing that we did is confirm that we see pain in these animals. So if you use von Frey hairs, you can test the amount of force that it takes for an animal to withdraw their paw in a painful response. And we can see that in EAE, you get a significant decrease in the amount of force required. So they're hypersensitive. So when we give a KOR agonist, we see an increase in this withdrawal threshold in the vehicle animals, as well as in the EAE animals. And there's actually no difference in the amount of analgesia between our controls and our EAEs. And that's true at both a low dose and a high dose of this KOR agonist. So our first question was, okay, well, do we see this same receptor decrease in the spinal cord as the last study? And we do, both in the mRNA and in the protein levels, we see a decrease in the kappa opioid receptor in our EAE animals. So if we see a decrease in the receptors, in the spinal cord specifically, then you would expect that giving the KOR agonist directly to the spinal cord, it's going to be less effective in EAE. However, that's not actually what we saw. there was no significant difference between the increases in the withdrawal thresholds for EAE versus vehicle. So why could that be the case? What's going on here? So the first thing we looked at was looking in the spinal cord to see the levels of inflammation that we could see, which again, that's one of our hallmarks of the disease. So you can see that in EAE, the microglia, as marked by IBA1, are significantly more activated. However, this was not always the case. There was a high degree of variability in the amount of inflammation that we saw in the spinal cords of these EAE animals, which actually correlates with the analgesia that we saw. So the more inflammation that we have, the less effective our KOR agonist is at giving analgesia. So very interesting, but it doesn't account for the fact that when we give it systemically, we see the same level of analgesia. So we wanted to look at KOR expression again, but this time in the brain. So we chose 3 regions here that are associated with both the kappa opioid system and with pain. So what we saw was that in the nucleus accumbens specifically, there was a massive increase in the amount of kappa opioid receptor protein. So there are a few different explanations that could account for this, but one possibility is that the receptor is being upregulated in this brain region to compensate for the downregulation in the spinal cord. So what do we know now? I've just put a little bit of a recap here of what I just said. I'll pull that back up for the discussion portion. But before I do that, I just wanted to thank both of the labs that I am a part of, as well as the lab members that have helped me on this project. And also thank you to CIHR and Alberta Innovits for funding my master's.
All right, thank you, Kaylin. And we have about 10 minutes for questions and answers. And thank you for leaving that slide on. So if any in the audience has any questions, please ask them now. Be a great moment. I have a question. I guess like this is kind of like the questions that most people get when they show animal models of anything. How does it translate across species? Basically, the specific manipulation that you interpreted here.
Yeah, that's a great question. Preclinical models tend to be a bit removed from the human population. However, we're using KOR agonists because they're currently being looked at as analgesics in the human population. So we have the same endogenous pain control systems in place. So it should be rather translatable there. Of course, more research is required. But yeah, and in terms of the model itself of MS, there's no perfect model because we don't actually know what causes MS, but the one that we use, as I said before, it has the same inflammation, autoimmunity, and demyelination that we see in MS.
A little pause here while we wait for questions from the attendees. I guess I'll follow up with another question, and this might be a little ignorant on my part. Can you say something about the extent of like that basically the EAE, the autoimmune encephalomyelitis, differentially affects in demyelination or inflammation. So which one is, all right, those two things correlated?
Yeah, so they do tend to be correlated. Yeah, so it's kind of a chicken and the egg kind of problem. Does the demyelination happen first or the inflammation? But you do kind of see them hand in hand. You see them together, and yeah, at our stage, we're not, the stage that we test the animals, we're not seeing as much demyelination, which is why we looked at inflammation here as a marker of disease severity.
I see. And in humans, my understanding was like, well, demyelination was like kind of like the first thing I think about when I think about MS. Is it that the case also in humans that you see inflammation before or like more pronounced than demyelination?
That is a good question. So it depends on the type of MS. So you can have relapsing remitting MS, where it kind of, you have time points where you get periods of inflammation and demyelination, and then it kind of goes away. And so it starts going back and forth, whereas you can have primary progressive MS, where it just starts getting worse, or secondary progressive MS, where it starts off as that relapsing remitting and then it slowly gets worse. And so again, yeah, the demyelination and inflammation tend to be highly correlated. You see them in the same periods of time. Oh, I think you're muted.
Thank you for your answers. So we have a few minutes. We haven't gotten questions from the audience yet, I don't think. Let's double check. But if anyone wants more information, I put the link to a recent article by Kelly on the chat. There's also her Twitter information. And if there's anything else, we have about 4 extra minutes here. Kelly, is there anything else you want to like say about your story, about the science that you haven't said yet because you didn't have enough time?
Yeah, if there's time, I might as well. So this is only actually one arm of the project that I'm working on. The other arm is looking at the effective side of the kappa opioid system. So the reason that that KOR agonists may be less addictive than mu opioid receptor agonists is because when the mu system is activated, you get analgesia, but you also get euphoria. So it makes you feel really good. And so that's one of the things playing into the fact that it's addictive. Whereas with the kappa opioid system, when it's activated, you get analgesia, but you also get dysphoria. So it makes you feel not good. So that's not ideal for a human population. So there are different ways that people are looking into fixing that. And so one of the arms of my project is looking at how the aversive component of this drug changes in MS. And what was actually really cool is that when we did a conditioned place aversion, so pairing the drug administration with a certain chamber, although in our controls, the drug was aversive, in our EAE animals, there was no difference in the time spent in the chambers. So they didn't find the drug as aversive. So that's really interesting. That's also another possible reason there are changes in the nucleus accumbens. That could be more factoring into that side of things. Yeah.
Oh, that's great. You said there was also, maybe not in the EAE animals, the addiction effect of the opioids. Have you had any behavioral studies trying to get at that too?
Yeah, so our lab, part of it focuses on MS. So we have another student, Zoe. She is looking at the mu opioid system in EAE. She's looked at the aversion as well, not quite at addiction, but we do have other projects going on looking at opioid addiction and withdrawal and changes in the mu-opioid system.
All right, that's great. So that's about the time we have for Kellen. Thank you again. Let me see, I have one more message here. And unfortunately, our third speaker has not shown up. So it might be the end of the session. But thank you, Kaylin, again.
Thank you.
And so if our host, I don't know if you want to put another screen saying that the speaker didn't show for the rest of the time.
I think we can just go ahead and shut it down.
Okay. All right.
But
Thank you,
Kaylin.
Thank you.
Go on first.
Yes, thanks, Heidi. So I'm Christine Charvet. I'm an assistant professor at Delaware State University, and I'd like to thank the organizers for setting up this meeting. And I'm excited to share with you how the integration across structural and transcriptomic scales moves us forward in studying development programs across species. And I'm presenting a preprint that's available on bioRxiv. So we study the evolution of the human brain. Here you can see a phylogeny of humans, non-human primates, as well as rodents. And we compare the brains of these various species because it's interesting to think about the evolution of the nervous system. But it's also really important for biomedical fields because we often use model organisms such as mice, rats, or macaques, and there is a need to translate findings from model organisms onto humans. And I'll talk about specific tools that we've created that enable researchers to map findings from model systems onto humans. And I'll talk about how the integration of structural and transcriptomic scales makes for better tools to study development programs in different species. So we focus on the developmental basis of variation in phenotypes across mammals. We start off life as a single cell. Cells proliferate and differentiate into diverse cell types. And we focus on conservation and variation in development programs as a means to understand the evolution of the human brain. And there are many metrics to compare when we talk about brain development, but we focus especially on the timing of development programs. For instance, by that I mean how long it takes for cells to be produced or how long it takes for connections to form. And there are two main reasons for focusing on timing. One is that it enables us to find corresponding ages across species. And that's something that's really important if we want to identify modified development programs in different species. And another major reason is because modifications to the timing of development programs have been a major driving force in generating phenotypic variation. So I've studied developmental timing for a long time at the structural level. And to move forward in the field, I trained in neuroimaging at Boston Children's Hospital and then in the Department of Molecular Biology and Genetics at Cornell University. And what I noted in each of these fields is that the tools available are really innovative to enhance our understanding of the brain, but the tools in each of these disciplines come with particular sets of limitations. And what I'm excited to show you is the combination of genetics and neuroimaging becomes this really powerful approach to study the evolution of development programs. So I'll talk about our past efforts where we've focused on the timing of biological programs to study corresponding ages during fetal stages of development, and how moving forward, the integration of neuroimaging with gene expression enables us to find corresponding ages across the lifespan. And with it, we are developing new tools to better study the evolution and development of connections. So in comparing brains of different species, one of the most obvious issues to address is that species vary widely in their length of development. As you can see here, humans develop prenatally for 9 months, but mice develop prenatally for 3 weeks. And for a long time, it wasn't clear how to compare brains of different species because there was no systematic way to find corresponding time points. But my collaborators and I developed a standardized approach. We collected a broad range of transformations, a few examples of which are shown here. And by collecting a broad range of transformations throughout development and across multiple species, we could find corresponding time points throughout development. And we could identify whether some development programs occur for an unusually long or short period of time in select species. This information is really valuable. So with this information, we built a website called translatingtime.org. And on this website, the user can find corresponding ages for any of the 18 mammalian species that we had studied. And this is a project which we call the Translating Time Project. And it's a project that started with the publication by Finley and Darlington in 1995, where they published a paper in Science. And it has grown immensely over the past 25 years. For a long time, we collected the timing of abrupt transformations. By that, I mean when eyes open, when cells stop proliferating to become neurons. And over the years, we had collected an impressive 354 transformations. And abrupt transformations largely occur during fetal stages of development. So the data set was mostly restricted to prenatal time points. And these kinds of data are available mostly for model systems. And we had collected data for an impressive 19 species. In my lab, we employ the multi-scale approach where we harness temporal variation in transcription, behavior, and neuroimaging. Now we work with data that's larger by an order of magnitude. For instance, we consider temporal variation in transcription for 20,000 plus genes, and that's just one example. Now we can find corresponding ages across the lifespan, and we can move beyond the model system. And I'll show you some interesting data where we can find corresponding ages between humans and chimpanzees throughout the lifespan, something that would have been really hard to do with the past approach. And we started to employ this approach, harnessing temporal variation in transcription to find corresponding time points at postnatal ages in humans and mice. We found corresponding ages up to 30 years of age in humans, which equates to 6 months of age in mice. And so if you study mice, I'd encourage you to check this paper out in cerebral cortex from the lab where you can find these corresponding time points. But in the interest of time, I'm not going to focus on this study. Instead, I'll focus on another study that really demonstrates the power of integrating across scales of biological organization. And it is one that involves a comparative analysis of humans and chimpanzees. Now, we often compare biological programs between these two species, often with the goal of identifying what is it about our biology that's different from one of our closest relatives. But we don't know how to map corresponding time points between these two species. And so I reason that if we could map timelines between humans and chimpanzees, we would be better equipped to identify modified development programs that might have been uniquely evolved in our lineage. And while the lab was getting set up, I collected different kinds of data, as you can see here, overlaid on a human timeline. So I considered behavior in beige, transcriptional variation in purple, and anatomical variation from structural MR and diffusion MR imaging. And what's of particular note is none of these metrics span the full lifespan. It's by harnessing these metrics together that we can find corresponding time points across the lifespan of these two species. Another major advantage for using these different metrics is that we can find corresponding time points from transcription and anatomy and compare the results. And by ensuring concordance of results across metrics, it makes for increased accuracy of the data. And this is something that's particularly important when we deal with human and chimpanzee data, which can often be variable and sparse. And so ensuring accuracy of results makes for accurate identified time points. I'll walk you through some examples. So here, I considered temporal variation in transcription from RNA sequencing data set. You can see that over time, there's temporal variation. And what I did is I aligned these trajectories and used those to extract corresponding time points. Here you can see these corresponding time points extracted from these alignments in humans on the y-axis and chimpanzees on the x-axis. Age is expressed in days after conception. And what I did is I extracted median time points from those data, and I used the median time points in the model moving forward. I also considered anatomical variations such as fractional anisotropy. And fractional anisotropy measures the alignment of fibers with a low value representing fibers that are not in alignment and a high value representing fibers that are in alignment. And what is relevant here is that there's temporal variation in fractional anisotropy. Here you can see fractional anisotropy varying over age in humans in red and chimpanzees in blue. And what I did is I aligned these trajectories to find corresponding time points. Here you can see a total of 137 time points collected for humans, as you can see on the y-axis, and chimpanzees on the x-axis. Age is expressed in days after conception. And what we notice is that time points extracted from transcription in purple, anatomy in blue, and behavior in beige align. They align so much so that a linear model through these data account for 94% of the variance. And what that means is that we can, with very good confidence, say how old a human is in chimpanzee days. Here are the data again represented on the right. And with these data, we can say that a 2-year-old chimpanzee is equivalent to a 2-year-old human. A 47-year-old chimpanzee is equivalent to a 55-year-old human. And so with this approach, we can find corresponding ages across the lifespan. So the integration across scales of biological organization is a powerful approach to translate time across humans and chimpanzees. It's not the only reason for wanting to integrate neuroimaging and gene expression. Major advantage also here is that we can use these tools to better study the evolution of circuits. And in the preprint, I integrate these tools to ask whether the development of frontal cortex circuitry is unusually extended in humans compared with chimpanzees, as had been claimed. But that problem is a complicated problem and one that deserves maybe more than a few minutes of attention. So what I will leave you with is this notion that by integrating across scales, and in particular neuroimaging and gene expression, the integration broadens the repertoire of tools to study biological programs, and in particular, the evolution and development of connections. And that this approach has already been quite powerful as it has served to find corresponding ages across the lifespan in humans and chimpanzees. And if you study chimpanzees, I encourage you to check out this preprint where you can map developmental timelines between humans and chimpanzees, which should yield a better identification of modified biological programs in the human lineage. I'd like to thank members of my lab, Melissa Harrington at Delaware State University, Barbara Finlay at Cornell, who started the Translating Time project, André, who generates these beautiful structural and diffusion MR scans, which we use in our laboratory. And I wanted to thank you for listening. I wanted to mention briefly that there are opportunities to join the lab, to join the lab and work part-time or full-time. You can join it in person or remotely. And if you're interested, you can email me at charvetcj@gmail.com. And if you want to learn more about our research, you can check out our website called charvetlab.com. And if you want to find corresponding ages across species, you can check out our website called translatingtime.org. And if you want to follow our updates, you can follow it on Twitter at CJCharvet. Thanks so much for listening.
Thank you. Wonderful. So is there any question? Okay. Yeah, there is. It seems that we have a question here in the Q&A.
I'll exit and stop sharing.
Hi, sorry. I guess as a panelist, I can't actually ask a question through the Q&A.
Yeah.
I'm wondering how sensitive this is to pointing out where there are, I guess, inconsistencies. Like if in the mouse, you see two different things that are pointing to two different ages that are developing, like the trajectories, I guess, of the developmental processes. Because the brain doesn't develop as one giant unit, like different parts go different. How easily can you pick that out with your methods?
Well, thank you for the question. So part of the advantage of using this approach is basically we can, map corresponding timelines between humans and mice and better compare them. And with that, we can identify developmental programs that might occur unusually early or late. And those would be reflected as sort of time points that are far away from the regression line. And those certainly do play a role in generating phenotypic variation. I didn't have much time to talk about it, but we focused extensively on the development of the cortex and looking at which developmental programs are unusually extended in primates compared with rodents. There are also, we noted some possible heterochronies in humans and chimpanzees as well. Particular weaning falls off the regression line, meaning there's a deviation in the timing of developmental program. But by collecting a large data set, we can then better identify which developmental programs occur unusually for an unusually long or short period of time. Thank you for that question.
Thank you. So we have 1 minute for another question. There is one in the Q&A. Would you mind to answer, Christine? Or do you read it to you?
Oh, in the Q&A, let me.
Yeah, I mean, the question is how can this age match account for variability inside a species like healthy humans?
Yes, yes, that's a great question. So what we've tried to do by collecting these different kinds of time points is reduce that variation, right? By integrating transcription and neuroimaging and averaging the data, we're hoping to find, the best match. But there's extensive variation within humans, and it's a problem. How can we get a good representation, of humans, etc. I don't have an answer. It's often the issue is just the data, what data we can capture. So it's a great question, and it would be really interesting to look at individual variation.
Okay, thank you.
Thank you.
Yeah, okay. I think we have to move on with our next speaker. So Kyla from Western University of Canada. So please.
Yes, hello.
Would you mind sharing your screen with us,
yes.
please?
All right. Okay, great. So I'm going to go ahead and assume that everyone can see my screen sharing.
Yes, we
okay,
do.
Great. Okay, so I am working on a project right now. Well, first of all, thank you for having me. I'm a graduate student at Western University, and I'm working on establishing a neurodevelopmental rat model with a specific focus on an area in the brain called the subplate and its role in the GABA system in the prefrontal cortex. Okay, so my model is trying to focus on specifically cognitive symptoms of schizophrenia. And so cognitive symptoms of schizophrenia are a predictor of functional outcomes. Schizophrenia traditionally has been viewed as one of the most functionally impaired psychiatric diseases or disorders. And of the 3 different types of symptoms or clusters of symptoms, positive, negative, and cognitive, cognitive symptoms have recently been gaining a lot of traction and focus because they really indicate or they're great predictors of not only functional outcomes, but also functional impairment in everyday life. And cognitive symptoms do have a significant interaction within the prefrontal cortex. And the GABA system has been identified as a key player in cognitive symptoms, as it may result in disrupted excitatory inhibitory balance, which may underlie these cognitive symptoms that we see within schizophrenia. And so the overall aim of my project is to, again, establish a neurodevelopmental rat model that show molecular abnormalities that are relevant to schizophrenia by lesioning this transient layer early during neurodevelopment called the subplate in the developing prefrontal cortex in rat. And so the subplate is a relatively novel field or area of research within neuroscience. And subplate neurons are one of the earliest generated and the mature subset of neurons. And they actually are just below the cortical plate. If you see here on the left, this is a P2 rat brain. And so I've outlined the cortical plate here and the subplate, as well as the white matter. So the subplate is very transient. In rat, it only stays around the first 1 or 2 postnatal weeks, and they quickly begin undergoing programmed apoptosis. And the role of the subplate during early development is to kind of serve as a waiting compartment. And so the thalamus during early development will project its thalamocortical inputs towards the cortical plate, and the subplate neurons will essentially kind of catch those inputs and relay them towards the cortical plate. And as I mentioned just a couple minutes ago, subplate neurons undergo programmed cell death towards the end of the first postnatal week, sometimes beginning of second postnatal week, really depending on the birth date of those neurons, which results in a more monosynaptic thalamic input to the cortex that we know are present in adults. And so here on the bottom left, there's a schematic, and layer 4 is just a layer of the cortical plate that one of the papers I really took inspiration from for my thesis was focusing on. And so that's why it just happens to be layer 4. But essentially, during early development, the thalamus will relay its inputs to the subplate, and the subplate neurons catch that and further relay it to the cortical plate. And throughout development, our subplate neurons undergo apoptosis, and it's a more monosynaptic thalamic input from the thalamus to the cortex. And so in lesioning the subplate, the hypothesis is that thalamic cortical afferent pathfinding may be aberrant, which may contribute to the development of abnormal cortical cellular distribution, potentially that we may see reflected in schizophrenia. And so these abnormal cellular distributions in schizophrenia include GABAergic neurons, and more specifically, a subset of those which are parvalbumin interneurons seem to be affected. And so literature consistently finds that there is a decrease in parvalbumin mRNA, which is layer specific, so layers 3 and 4 in the dorsolateral prefrontal cortex, as well as a decrease in protein levels in the dorsolateral prefrontal cortex as well. In addition to parvalbumin deficits, there also seems to be a consistent finding of aberrant GAD67 protein levels, specifically, again, in layers 3 to 5 dorsolateral prefrontal cortex. But also it's interesting to note that this decrease in GAD67 protein levels are actually only found in a subset of parvalbumin positive interneurons. And so GAD67 doesn't seem to be a universal deficit across all interneurons, but rather just a subset of parvalbumin positive interneurons within schizophrenia. And so in 2006, two researchers, Dr. Kennels and Dr. Schatz, did a very elegant study in the visual cortex of cats. And my thesis was actually very heavily inspired by this project. And so in their study, they lesioned the subplate in the visual cortex of cats, and they found that it resulted in an immature GABAergic system in adult cats. And a lot of the molecular abnormalities they found in these cats as an adult were very similar to molecular abnormalities that we see in postmortem schizophrenia brains. One of them was decreased KCC2 mRNA levels. KCC2 is a chloride cotransporter protein, which I will talk about in just a minute. And they found a very localized deficit in specifically layer 4 of the lesioned subplate brain as an adult. And they also found that perforated patch recordings revealed a depolarizing current only in layer 4. And so GABAergic neurons are traditionally hyperpolarizing. However, they found that a lesioned subplate in layer 4, they found it to be depolarizing. And so these chloride cotransporter proteins that I was just mentioning, one of them is KCC2, and KCC2 acts in tandem with another chloride cotransporter protein called NKCC1. And so early in the immature brain, NKCC1 is expressed more in relative to KCC2. And so the ratio of these two proteins that are expressed eventually determine the intracellular chloride concentration, which subsequently predict or affect the GABA A receptor function, which then determine the GABAergic response. And so in a nutshell, as the brain develops, neurons naturally express an increasing KCC2 amount and decreasing NKCC1. And so the ratio switches, which allows for our GABAergic response to change from one that is excitatory or depolarizing to hyperpolarizing. And so essentially what the study found was that in adults, the GABAergic system was remaining an immature one. Previously, my lab have also taken a look at these animals and done several behavioral tasks. And we have seen traditionally that the NGF lesioned animals or the subplate lesioned animals performed worse on several behavioral tasks that were related to focus and traditionally related to schizophrenia behavioral models. And so just for the sake of time, I'm just going to go straight into the methods. My project, we injected P1 rats bilaterally into the developing prefrontal cortex with NGF or sterile saline, and they were sacrificed in multiple time points from early postnatal weeks from P5 to adulthood. They were immersion fixed, and then coronal sections were cut and subsequently immunolabeled for KCC2, that chloride cotransporter protein, parvalbumin, and GAD67. And then images were analyzed with ImageJ software. And so I use NGF to lesion mice subplate, which essentially during early postnatal weeks binds to P75 receptor, causing cell death specifically within the subplate layer. So my study has found that this parvalbumin protein and GAD67 protein have a difference, or more specifically, sorry, the parvalbumin intensity it seems to be lower in NGF animals as compared to saline animals, which is our control animal. And so this is only specifically in the lower prelimbic cortex, which I have assigned as layers 5 and 6. We haven't seen any significant differences in the density of either parvalbumin or GAD67 positive cells, nor have we seen any significant differences in GAD67 fluorescence intensity levels. However, there seems to be a layer-specific parvalbumin intensity difference across our groups as we expected according to literature seen in postmortem schizophrenia studies. There we go. I also got a chance to look at KCC2, and so KCC2 just has a kind of fluorescence signal distribution pattern or expression pattern that is quite difficult to quantify in terms of how many cells or density of number of cells that are expressing KCC2. And so I just decided to stick with the intensity outputs. And I found, so the analysis shows that there is a significant difference in the intensity of KCC2 across our experimental and control groups, specifically in the upper prelimbic cortex. To my knowledge, there are no studies right now that look at KCC2 levels in the early postnatal weeks. Most studies only look at KCC2 as an adult. And interestingly, we didn't find any differences as an adult in KCC2 intensity. Another protein that I am slowly starting to analyze and take a look at is NKCC1, which again, as I mentioned earlier, goes kind of, is kind of partnered with KCC2. And so I thought it might be just nice to show you the distribution patterns of NKCC1 in a control animal early during postnatal weeks. So this is postnatal day 2 when NKCC1 is expected to be expressed at high amounts. And you can kind of just see the outline of the cell membrane here within the cortical plate. And so to conclude, there's a decreasing trend in parvalbumin protein expression in subplate lesioned rats in the prefrontal cortex in line with several postmortem schizophrenia studies. We are seeing higher KCC2 expression in early postnatal NGF rats compared to their saline counterparts. This could be due to the subplate's role and KCC2's role in neuronal migration. And so this could be a compensatory mechanism, and NKCC1 expression patterns are to be determined in subplate lesion rats.
Great, thank you. So we have an open question for you. What about if we just move on with our next speaker, and you could write an answer for
sure.
attendee? Okay, thank you. So Tsutsi from University of Toronto. She's gonna talk about autism in mice and humans. Please go ahead.
Hi, can you hear me?
Yes.
I forgot to unmute myself. Okay, hi. So I'm going to be talking about trials and tribulations, co-clinical trials of autism in mice and humans. Basic introduction to autism. It's a neurodevelopmental disorder characterized by impaired social interaction communication and restrictive repetitive behaviors. It has a strong genetic basis, but it's extremely heterogeneous, which unfortunately leads to issues when it comes to treatment. So one of the analogies that I like to use is another disorder that's very heterogeneous, which is cancer. So imagine if we took 100 people with cancers, gave them a drug. Of course, you're not going to see any treatment response. And that's essentially what we're doing with autism right now. We're taking 100 people with a very heterogeneous disorder and treating them, and it's not working. You have to figure out, in the case of the cancers, who has breast cancer, lymphoma, brain cancer, prostate cancer, lung cancer, etc. And even when you take out those individuals with lung cancer, you still might not be seeing treatment response because it's so heterogeneous. And that's actually what happened in the study that is the motivation for my PhD thesis project, where they did a study in lung cancer and found no treatment response, but they implemented this novel idea of doing a co-clinical project, which is simultaneously treating preclinical models, so mouse models of that cancer with the same medication. And they found that depending on the mutations within the preclinical models, they actually were seeing a treatment response. And then they used that information to inform their clinical trial and found that a subset of the individuals were in fact responding to the treatment, but it was getting averaged out over the whole population. So we want to take this concept of the co-clinical project and bring it to autism to see if we can help attack this heterogeneity issue and subtype within autism. So the overarching project is to take this co-clinical project, the clinical population and the preclinical population and treat them with 3 different drugs that are promising for autism, assess behaviors and imaging, and then predict responders and non-responders to subtype autism and have a more specialized, individualized treatment approach. I won't go into too much details here. I actually want to focus more on the overarching method of the co-clinical project, but we did use 3 different drugs. So in the humans, there were phase 2 studies with all 3 of these drugs. The oxytocin and tidaglutide studies have actually been completed, and our baclofen trial is ongoing. For the mouse side of things, which is the side that I was in charge of, we started treating the mice around 5 weeks of age, and before we started, we used manganese enhanced MRI imaging to assess their structural neuroanatomy, and we had multiple time points of that to see how their neuroanatomy changed over time. The treatment was in a method that's similar to the humans, so we were doing intranasal drops onto their nose. And we also looked at behavioral testing with various behavioral metrics to assess if we had some sort of treatment response. We modeled this to reflect the human trial to the best of our abilities, and we used 3 different autism-related models for the study. Again, I'm not going to go into details. This paper is actually on its way out. I'm happy to share the results if someone is interested, but overall, we found little to no effect of oxytocin, neither in the behaviors nor in the neuroanatomy. And so we sort of were thinking, is this something that we did? Did we not assess the right measures? Did we not do this test the best way possible? And what can we change moving forward to make sure that we do capture treatment response? So for the next drug, we changed a few things, like we put all the mice on the same background to increase our power and actually increase the number of models we were looking at. We added imaging time points. We changed over the behavioral measures so that we had replicability. So now we have 2 measures for anxiety and hyperactivity and 2 measures for learning and memory to make sure that we're actually capturing the behavior that we're interested in. Even still, we found subtle and negative effects on mouse behavior with this drug. So you can see in the top left of the OMAs measure of anxiety, one of the mouse models actually had a decreased anxiety to abnormal levels, and nothing else particularly like interesting or exciting. One thing we did find was it was not a measure that we were interested in looking at, but we looked at sleep activity because it was sort of collected as we were going. And there seems to be a trend in treatment actually affecting sleep. But again, we weren't actually like assessing this, but this is something that we considered moving forward. We also found no effect on mouse neuroanatomy with this drug. So here you're looking at coronal sections like through the mouse brain. And anything that is blue are smaller relative to controls, and red or like yellower is larger relative to controls. So in the middle section where you see strain differences, we do see expected strain differences by the genotype, but we were not seeing treatment effects. So if you look at the leftmost column, you see there's no colors on that column, and that's because we didn't see any changes. And even though it's the treatment in wild type, this column is actually representative for all the strains. We did see, if we brought the threshold to an FDR correction of 20%, a very subtle interaction effect in the ARID1Bs. So on the rightmost column, you can see that. However, if you like look, you'll see that it's so subtle that it's very hard to actually see them in the figure. So we wanted to see whether maybe our lack of treatment effect was due to we just have too many variables. We're not going about assessing it the best way. Maybe if we reduce our dimensions, if we use a multivariate approach, we can actually predict responders and non-responders across the species. So in the top row, you can see the mouse PCA. So we use principal component analysis, breaking down by brain as well as brain and behavior. And the bottom row, you can see humans. And blue is the treated individuals and pink is the non-treated, like the controls. And as you can see, there's nothing that particularly stood out. There's no clusters here or there. That being said, when you look at the human data, our sample size is incredibly small. You can imagine the difficulties that come with imaging children, just in general, but especially children with autism. So we only have a sample of 8 controls and 5 tidaglutide treated individuals. But if you look at the graph, there is and I don't want to use the word cluster here because the sample is far too small to actually say that, but there is a collection of points. So these are individuals that are grouping together, which shows some sort of promise for using this method, but without a larger sample size, we cannot say anything conclusive about this. So our next drug, we're going to be using our baclofen. And as you can see, we've changed a lot more things. We want to make sure that we're actually doing this like as completely as possible. So we've increased the number of strains we've had. We've increased the number of behavioral tests, including doing a pre-behavioral test. We've changed the mode of administration to drinking water instead of IP or the nasal to create an environment that's less stressful. So we're not going in every day and injecting these mice. We've added the home cage monitoring as an actual measure that we're interested in assessing because that was one of the measures that I talked about earlier, where we saw an effect on sleep, but because we weren't powered to actually extract anything from that, all we can say is we saw something. We've also added microbiome collection, again, so that we're approaching this as thoroughly as possible. The one thing I will say is we are in the process of piloting switching our method over to IP injections instead of drinking water. We've been having issues with the mice being incredibly aggressive, and we're not entirely sure whether that's from the drug or the drinking water or what exactly is happening there. So we're comparing the two methods. So our initial expectation for this project was take a bunch of humans, take a bunch of mice, treat them with medication, and then get this wonderful ability to predict responders and non-responders. And as science always goes, the reality was not that at all. And there's a lot of things I can say in regards to limitations. Some of the things are probably more obvious. I'm not going to go into them just for the sake of time. But one thing I do want to, I guess, talk about a tiny bit is the concept of responders. So we want to get to this idea of how do we separate out who is a responder and a non-responder. But in autism, it's not as straightforward as it is in cancer, which was the proof of concept for this project, the co-clinical project. So in cancer, you have just a tumor, and if that tumor shrinks, then you're happy. In autism, that's not how the disorder is defined. So how exactly do you know, like, yes, they're responding? And one of the examples that's really great for this is there was actually a clinical trial where they used arbaclofen, actually. And overall, the treatment didn't seem to do anything on their primary measure, secondary, anything. But afterwards, when they talked to the participants and their parents, it was very obvious that when they chose which treatment that they wanted the treatment, the actual treatment, not the placebo, but this didn't come across in the clinical trial itself. Okay. There's a couple of other things that I could go into, but I have a feeling I'm running out of time. So I'll just leave those there, and I'm happy to talk about it if anybody wants to later. I'd like to thank a ton of people and organizations, and thank you all for listening.
Thank you very much. So it was a wonderful talk. We have time for one open questions. There is one in the Q&A. You go, please.
Okay, I see. So can you say how you made the autism mouse model, and does it mean that the drugs you mentioned were simultaneously being tested on humans? Yes. So the drugs that were being used were more or less simultaneously being tested in humans. So there are 3 clinical trials that are sort of associated with my thesis project. And the autism mouse models, there's a ton of models that are, I guess, well known or well used in the field. These are fairly well studied, and there's quite a bit of literature looking at like how reliable are they, like construct validity, face validity, etc. The ones that we used, we ended up primarily using ones that were highly associated with autism. So we didn't actually make them; they already exist. You can go to the Jax or whichever your favorite mouse model company and buy the mice as they are. Yeah, happy to talk about that more if anybody would like to.
Great, thank you. So is there any other question? We have maybe 1 minute for another one, or any comment you wanna make? Okay, doesn't seem to be the case. I just wanna thank you, all of you, the wonderful speakers, the audience, and of course our backend. He was making sure that everything here worked fine. So good luck with your projects, and see you next time.
Thank you. Thanks a lot.
Bye.
Hey, Ramon.
Sorry, I was muted. Hi, how are you? Nice to meet you, Nidhi.
Yeah, nice meeting you. Looking forward to your talk.
Thank you, yeah, and thanks for your time organizing
No
this.
worries. I look forward to it. Maybe I can introduce you to Shreya, our backend host. Shreya, do you want to say hi?
Yeah, so we talked already and he shared his screen and everything, so we're good.
Okay, great.
Yeah.
Just to remind, like, yeah, you sent me an email with all the info, but just to remind me, so Nidhi, you're gonna be like the front person, and Shreya, you're gonna be helping in the back. But I forgot what you were gonna ask the questions, or who I missed that part. So what's the different roles between the two?
So Shreya is basically just gonna take care of like the Zoom and letting people in and things like that.
Right.
Like the technical stuff, and I'll be basically asking you questions on behalf of the attendees that they post in the Q&A, and also I'll do a brief introduction. For the past few sessions that I emceed, I've been saying the title of the talk, but I think I'm going to stop doing that because when I say that, the speaker also repeats it, so it becomes like a waste of time. So I'll just say your name and your affiliation.
Okay, makes sense. Yeah, I'm gonna in my guide notes, I was supposed to say the title myself. So yeah, maybe it's better if I just say it myself. But
Yeah,
yeah.
sense. And also
Cool.
like the titles can be long and I don't want to mess them up. So
right.
So actually your last name is Ramon Nagera.
Yeah, it's when I'm from Spain, and when I left Spain, I realized it's not easy to pronounce. So the actual pronunciation, but I don't have a strong opinion for it. It's Nogueira.
Nogueira. Okay, got it.
Nogueira, yeah. But don't worry. If you don't say it exactly perfect, it's zero problem. Really, don't worry.
Awesome. Yeah. Hi, Luisa.
Hey, Nidhi. Did you get my email? My internet just went down, so I'm using my phone. So please let me know if my internet is lagging or anything, okay?
You seem fine right now. Do you want to try sharing your screen?
Yes. Okay, let's see. Okay, can you see it?
Yes.
Wonderful.
Cool. If either of you have videos, you can try to see if that plays, but if not, that's fine.
Oh yeah, I do have a video, so I'm gonna try.
Okay, sure.
Should I share my screen?
Yeah.
Wait, what's happening? Oh.
Yeah, that's good.
All right. Perfect.
Awesome.
Okay. I also have a video, so let me make sure that it does work.
Yeah.
Let's see, PowerPoint. Okay, can you see it?
Yeah.
Awesome.
So it's gonna be 3 or 4 speakers?
3. We're
3.
waiting on one more actually.
Right.
Let me just.
By the way, hi, nice to meet you, Luisa Schuster.
Hi, Ramon. How are you?
Good. How are you?
Good.
But yeah, we'll go live in like 6 minutes now. I think Sarah is the other one, right?
Thank you.
Yeah, yes.
Is she first?
She is, yeah, she's the first speaker.
have a question regarding the format. So I chose the interactive talk. So I go for 5 minutes, and then what happens for the next 10 minutes?
Going to be a discussion for the next 10 minutes. I was actually just going to bring that up, so I'm glad you did.
Okay.
Yeah, so you'll present, and then we'll just open it up for discussion and Q&A for the remaining 10 minutes. Yeah, I think it's something about the online format, but people like tend to forget that they're presenting. So I'm always mentally prepared for people to not show up. I just wrote to Sarah, the other speaker.
So it's happened before that people just didn't show up and things have to be rescheduled somehow or not.
Yeah, that happened like a few times, not too many times, but I think it's something about the online format that people just tend to like have misscheduling. Maybe it has to do with time zones and stuff.
Right.
Yeah. Shreya?
Yeah.
Do you mind? My phone just conked off and I don't have like the Neuromatch Slack on my desktop. Do you mind just writing on the showrunners that our speaker who's supposed to go at 5 is missing?
Okay, yeah.
Yeah, I don't mind going first if that's the case. And I'm the second one, right? So I can just go first.
Yeah, I think that would be fine. But the only issue is because it's like a multi-track conference, some people who want to listen to your talk might show up, only for yours. So ideally, we don't want to move you around.
Okay, makes sense.
Just in case someone specifically wants to hear yours or something. Cool. But I'll just, in case, I'll make a slide saying, waiting for the speaker so that we can pull up the slide.
Okay, I'm going to start the webinar so that people can start coming in.
Just give me 1 second. Yeah, you can do that. I have a slide to present in case.
Okay.
Yeah, okay.
Perfect.
Cool, I guess we are live now. Hey everyone, welcome to this session. We are currently waiting for our first speaker to show up, and we don't want to start with the next speaker in the meanwhile because we know some attendees might show up specifically for the second talk. So in the meantime, we're just going to wait to see if she arrives. So please feel free to wait with us for Sarah to come for her presentation, or you can come back at 5:15 p.m. for the next two presenters. Oops, I just edit that. Hi. Hey everyone. So looks like our first speaker is here. Hi Sarah.
Hello.
So I think you'll have to give a slightly shorter talk than you had originally planned. So let's go ahead and maybe take, can you give it in 4 minutes so that you can have some time for question and answers?
I'll try my best. Sorry, I had some connection problems.
Oh, no, that's fine. No worries. So I'm not going to spend any time on the introduction. This is Sarah Seo. She's a PhD student at York University. Take it away.
Okay, thank you. All right, so I'm actually a undergraduate psychology student at York University, and this presentation today will be on the visuomotor transformations and the multi-unit activity of the frontal and supplementary eye fields in rhesus monkeys. So basically in systems neuroscience, it's a central goal to relate the neuronal activity to behavior. And here during a Q conflict memory guided visuomotor saccade task, it was performed by head unrestrained monkeys. We recorded some spiking activity from the frontal eye field neurons to examine if the multi-unit activity carried the same information as the single unit activity. And single unit activity is isolated from the multi-unit activity, which refers to the average firing of neurons recorded within the vicinity of the microelectrode. And here is an example of two isolated single unit activities, one in pink and one in blue. And it's also corresponded to the colored waveforms. And however, because neurons work in coordination with one another, there has been an increase in trend toward analyzing multi-unit activity instead. And so here we decluster the single units by re-thresholding the neural signal to obtain the multi-unit activity, as you can see in the yellow cluster, and we remove the unwanted noise, which is the gray cluster as seen here. So here's a simulation of the task with the timestamp located on the top right-hand corner. The monkey began the task by fixating on a red dot, as you can see in the middle of the screen, while a landmark was present on the screen. Next, a target was flashed. And this is what a visually responsive neuron would actually respond to. Then there was a delay, a mask, and a landmark shift to, for example, the top left-hand corner. So this was done pseudo-randomly, one by one. And then the monkey was cued by the disappearance of the fixation dot to saccade toward the remembered target. And here we recorded neurons that responded to the movement of the eye, which is also called motor neurons. And the monkey was rewarded for looking at the virtually shifted target, which is T prime as shown on the screen, target T or somewhere in between. And we tested the response fields against spatial models to reveal the codes embedded in the visual, memory, and motor multi-unit activity. And our previous findings have shown that the visual response codes best for target relative to eye, while the motor response codes best for future gaze relative to eye. But to investigate where exactly these codes or the response fields fall relative to the target or the gaze, we plotted a target to gaze continuum, which has 10 steps in between. But what exactly is a TG continuum? It's essentially the space between the error between the target and where the monkey's gaze landed, as indicated by the brown arrow right over there. And just to give a little bit of insight as to how we actually fit these spatial models, if activity is plotted in the correct reference frame or the coordinate, it will result in lower residuals compared to when the activity is plotted in the incorrect coordinate. So the correct one is shown on the left-hand side in this example with the lower residuals. So here are some exemplar or typical visual response fields and the dissociation between the spatial models. Or the visual and motor response fields against the TG continuum. So for the visual response, along the x-axis is the time frame, y is the frequency, and each red dot represents an action potential, and the red curve is the spike density plot, and the red shaded area is a sampling window of analysis. The blue line at time 0 milliseconds is target onset. And here's a single unit visual response field along the TG continuum. Along the X and Y axis is their horizontal and vertical dimensions, respectively, and each circle represents the spiking activity. The bigger the circle, the greater the magnitude of the activity. And the color map shown on the right corresponds to the non-parametric fit to these data. The red-yellow blob represents the hotspot.
As indicated by the dotted lines, the visual response field fits exactly at T in the TG continuum. And for the multi-unit response field from the same site, note that the color map depicts a higher firing frequency since a multi-unit consists of many neurons. The multi-unit visual response field is comparable to that of the single unit, which also fits exactly at T and also has the same hotspot. In terms of the motor response, here's the means by density plot aligned to saccade onset at time 0 milliseconds. And the single unit motor response field fits one step away from G, while the multi-unit fits 2 steps away. So overall, these specific examples show that the multi-unit activity carries the same information as a single unit activity. And lastly, quantifying it at a more populational level along the TG continuum and neuron count of the y-axis. First, for the visual response, as indicated by the blue, by the dotted pink line, which is the median, the populational distribution again fit best at around T for the single unit, as well as for the multi-unit. And that goes the same for the motor response, where the median you can see is closer to G for the single unit, as well as the multi-unit. So basically, in summary, similar to the single unit response, the multi-unit visual response fit best coded for target in eye and multi-unit for future gaze and eye coordinates. The results suggest that the multi-unit activity carries robust information for the visual motor transformations and therefore can be used as a reliable marker for those transformations for translational purposes in medical settings when, for example, analyzing single unit activity proves to be non-pragmatic. And that would
thank you for your talk, Sarah. Let's see if there are any questions. Let's give people maybe a few seconds to write their questions. Okay, so let's move on to the next speaker since it's 5:15. So our next speaker is Louisa Schuster. She's going to give an interactive talk. So that'll be 5 minutes of speaking and then 10 minutes for discussion. She's currently a grad student at NYU working with Robert Frumke and Adam Marr. So over to you.
Hi, everybody. I'm having some connectivity issues, so I'm going to turn off my video because I'm using my phone hotspot. So sorry that you won't be able to see my face, but I'm going to start my talk, okay? Let's see. Okay, I hope that you all can see my slide. As Nise said, I'm Luisa. I'm interested in studying maternal behavior and more specifically oxytocin and how that modulates maternal behavior. And today I'm going to talk about my project and what I've been doing to get to a place where we can ethologically assess maternal behavior in mice while still doing neural recordings. Maternal behavior is essential for life. It's the first social interaction an animal experiences. It's expressed across mammals. And because it's robust and reliable, it allows us to make behavioral assessments that are straightforward and enable us to study the connection between neural circuit function in vivo to behavior. You know, from this picture, from mice to humans, animals, mammals engage in maternal behavior. I study mice and maternal behavior. And one of the most robust and reliable maternal behaviors that we see in mice are pup-retrieval behavior. If you work with mice, you probably have experienced these where when a mom leaves the nest, she might accidentally drop the pup that is attached to her nipple, and the pup outside of the nest starts making these vocalizations. They're very vocal, and mom goes out, picks up the pup, brings it back to the nest. So this is a new mom. I'm showing you a video of a mom that she had just given birth a couple of hours before, and this is the first time that she's retrieving a pup. Previous work in our lab showed that, just like in the video, a mom that hears a pup that has been isolated from the nest will retrieve it automatically. This is a spectrogram of the ultrasonic vocalizations that the pups make when they're outside of the nest. So you get the auditory stimulus as a mom, you hear the pup, you go pick it up. However, a naive virgin, so a female that has never been exposed to pups or has given birth, when she hears this pup, she does nothing. In fact, you might find it so annoying that it starts grooming, self-soothing. A previous grad student in our lab, Bianca Jones-Marley, who's now a PI at Columbia, she discovered that if you co-house these naive virgins with moms and pups, and you give them oxytocin, this causes the virgins to start showing this maternal behavior. So what's oxytocin? Oxytocin is a neuropeptide that is synthesized in the hypothalamus into nuclei, the paraventricular nucleus and the supraoptic nucleus. And the PVN has outputs into the auditory cortex. And in this graph that I'm showing you in the right is what I was telling you that Bianca discovered, that if you give these animals oxytocin, whether it's optogenetically or systemically with an IP injection, this will make the acquisition of this pup retrieval behavior faster. So pup retrieval behavior is just one of the many maternal behaviors that mice show and mammals show. So we did another experiment where we thought, well, how are these virgins acquiring this maternal behavior? Because it's not magically like you're turning on a switch with the oxytocin. What are the behaviorally relevant episodes that are allowing this virgin to show maternal behavior? So what we did, this was under the supervision of Joanna Karsha, who's now a PI at Rutgers, was we created a behavioral monitoring system that allows us to look at 4 to 7 days of 24/7 behavior and all the interactions that were happening between a mom, the pups, and a virgin. And we look at neural signals in the brain of the virgin and how she was learning. And thanks to this more long-term behavioral monitoring system, we discovered that a couple of cool things. The dams learn to shepherd the virgins to the nest. And also, when the virgins are observing a mom that is retrieving a pup, like in the panels on the left that I have, it's just a sequence of a dam that is picking up the pup while the virgin watches. This is activating the PVN oxytocin neurons of this virgin. And we wouldn't have known this if we didn't have these 24/7 observations of these interactions that were happening in the maternal cage. So for my PhD, what I've been working on, or what I want to know is, okay, a mom is first a virgin and then becomes a mom, but what's happening naturally? What's the trajectory of oxytocin over time? Are moms becoming better the more leaders that they have as they become more experienced? How is this happening developmentally? How is the PVN modulating cortex? And more importantly, what are these behaviorally relevant episodes of interest? So I'm planning on doing a neural documentary on oxytocin where I do recordings from when an animal is a virgin all the way across 3 consecutive pregnancies. But in order to do that, I needed to create a system that was ethologically valid, that allowed me to see behavior in real time in an ecologically valid, in a niche valid way, that where I could integrate behavioral neural data, and that allowed me to have consecutive rounds of breeding. And also that the output allowed me to phenotype social interactions. So I built a box, a maternal enclosure, using Raspberry Pi computers, a couple with Raspberry Pi cams that allow you to see the nest from above and from the side. I also included an intake and an exhaust fan to control temperature, humidity, ventilation that are connected to an Arduino board. And the Arduino board receives information from a couple of sensors, a temperature and a humidity sensor, and it adjusts the revolutions of the fan in order to maintain the thermal comfort of the animal. I also included some LED strips that simulate the sunrise and the sunset, and I've added infrared LED rings in order to have night vision of these animals. And also an ultrasonic microphone, because the pop calls are important to us. They are what is triggering this mom to engage in certain maternal behaviors. So all of these, my plan is to make it open source for everybody. It costs around $150 to make it, except the ultrasonic mic. But we're trying to build also inexpensive ultrasonic mics so that labs, whether they are in the United States, Europe, Latin America, are able to build these open source enclosures. So I just want to show you a couple of things that we've found. So these are some of the behaviors I've been coding while these animals are in the enclosure. I'm using the ethogram provided in mousebehavior.org, is where the consensus of mouse behavior ethograms is. And I'm going to show you a couple of ethograms that I made, showing the behaviors that are highlighted here. And before that, I want to show you that one of the cool things about this enclosure is that you can get really up close to the nest. Instead of having just the bird's eye view of what your mouse is doing, you actually can see if your mouse is grooming the pups, which pup is getting fed, etc. So this is in great detail. So let me show you. So I've been hand coding these videos. Lots of work using data view software. If you need to annotate videos, I do recommend this software. It's excellent if you have very long videos, and it has great support. It was developed at NYU by the Adolf Lab. And in this slide, I'm just showing you an example of a virgin that is housed alone. So there's no mom, there's nothing. It's just a female mouse, a young female mouse. And this is just showing you some of the activity that this mouse engages in for 5 days, depending on the light cycles. So lights on, light off. And for example, what's interesting, if you look at day 1, is that as soon as you turn the lights off, in orange, you can see that this mouse starts being really active and build the nest. Okay, and some of the ethograms. This is an example of an ethogram of a mouse, 3 liters. And these are the first 3 days when the pups are born, and of 3 consecutive litters. That also shows you the richness of behaviors that these mice are engaged in. So I wanted to make sure that I created an enclosure and also a pipeline to be able to process this data before I go into the neural data. So since it's a 5-minute talk, I'm not going to get into details and patterns, but I would like suggestions if you can think of great ways to be able to represent this data and to analyze it, I would love to hear it, because we are collecting video 24/7, we're collecting audio 24/7, and ethograms are just the beginning, but we might be able to see other patterns. So please let me know what you think of it. But something cool that
since
yes.
you have only a couple of minutes, why don't you wrap it up and take one? You have one question, so.
Okay, yeah, I'm about to, like, I just wanted to show you this, for example, a pattern of one of the mice that we recorded from, and as time goes by, the more litters that these mice had, the more time it's spent in the nest, and therefore less time out of nest. And the nest that they were building became better and better. So in the future, we want to automate behavioral scoring. We're working on using deep lab cut. And we're starting with the acquisition of neural data, which is going to be fiber photometry in the beginning to look at the patterns of activation of PBN. And now I'm going to look at the questions.
Yeah, so the first question, I think you already answered that. So I'm going to encourage you to skip that one. It's just basically asking you, how you did say you're going to use deep lab cut, so that's fine, but maybe you can answer the second question.
Okay. So what type of camera is it and which specification does it have? The camera that we're using, we're actually using the Raspberry Pi cameras, and they're like 10, 15 bucks. So if you just look up Raspberry Pi cam, that's what we're using. And it's a wide angle lens, and you can really see up close, and it's really fantastic and very cost effective.
Cool. Thanks for the talk. I do have one more question. Actually, do you think you'll be able to differentiate between the pups, like their identities, or will that matter for your study?
That's an excellent question. So we've tried a couple of things. The thing is that when the mother hovers over the pups and when the mom groups them, whatever mark that you make either disappears or the mom licks it up. So we're trying to use deep lab cut in order to do that. We've used other strategies like, markers and stuff, but that's not good enough because if the mark is occluded, you cannot see it. But having the view from a couple of cameras really, really helps. Okay. So if that's where we're at.
Okay, great. Thanks a lot. If you can unshare your screen, then maybe Ramon can share his. In the meanwhile, I will introduce him. Our next speaker is Ramon Nogueira. He's a postdoc at the Center for Theoretical Neuroscience at Columbia, working with Stefano Fiusi. Take it away, Ramon. Your mic is muted.
Hi, can you hear me now?
Yes.
Okay, yeah, sorry. All right, so good afternoon, and thanks for being here, and thanks for Neuromatch and Shreya and Nidhi for organizing this. So today I'm gonna talk about, sorry, one second. I'm gonna talk about high-dimensional neural representations for low-dimensional tasks, a compromise between complex discrimination and generalization. This project is a collaboration between Stefano Fiusi Lab and the Randy Bruno Lab at Columbia University. And importantly, all the behavioral and electrophysiological data that I'm going to present today has been collected by Chris Rogers. We already have a first manuscript of this study, which can be found on bioRxiv. I think maybe Nidhi is gonna send the link on the group chat. So what I'm going to, or I can send it afterwards. But what I'm going to present today builds up upon this first manuscript. All right, so let me start with a brief introduction. As we all have experienced, the sense of touch can be very informative about the external world. Mammals, and in particular primates, can, for instance, use their hands or limbs to identify properties of objects in their surroundings. Even though we lack an intuition for it, other animals like rodents can likewise use their whiskers to recognize their close environment and discriminate, for instance, between a solid wall or a wall with a small hole. In this study, we aim at characterizing what are the complexity properties and neural representations of whisker-based discrimination tasks. Or in other words, can the behavioral task be solved by a linear or nonlinear integration of sensory cues? And are the neural representations in the somatosensory cortex high-dimensional or low-dimensional? So in this study, head-fixed mice were trained to perform a discrimination task by whisking in the dark. By whisking in the dark. They were asked to discriminate between a concave and a convex shape by freely sampling these objects for 2 seconds. And in particular, the time course of the trial was the following. A concave or a convex shape, a convex or a concave shape, started moving towards the animal at t equals -2 seconds. And this shape would start at 3 possible distances, the far, medium, or close. And at t equals 0, the animal had to make a choice by leaking on the left socket or for concave shapes and on the right socket for convex shapes. Importantly, animals were free to leak during the whole trial, but the choice at the particular trial was considered to be the first leak after this response window opened at t equals 0, right? Also, the position of these whiskers and the shape was recorded with an ultra-fast camera, and they were tracked on each time frame with DeepCut which I guess many of you are or have heard about. So if you're interested in all the exact details about this particular task, check this manuscript, bioRxiv, Rogers et al. 2020. And all right, so let's have a look at the video. It's going to be much easier to get a grasp of the task in particular. For instance, we have a convex shape here. The animal is making the contacts. Also, we can have a look at what happened. Oh, sorry. Oh, sorry about that. Let's have a look at the concave shape now. Right, so now we have a concave shape. Same, animal whisks, and the whiskers make some contacts, and which is going to be used to decide whether that was a convex or a concave shape. All right, so let's look at some behavioral results. So in this plot, we have the mean contact rate as a function of time across animals for all the whiskers. And in the beginning, where the shape is not present, is out of reach, there's no contacts. But it is between -1 and -0.5 seconds where the biggest majority, the majority of the contacts happen. And yeah, we have the different whiskers. So as I said before, animals are free to leak throughout the trial. Therefore, we can plot the probability of making a leak on the correct side as a function of time. In the beginning, when there are no contacts, the animal makes random choices, right? 50% performance. Nevertheless, once they are able to sample this sensory information by whisking, we see a performance increase. And at the end of the trial, this performance, the overall performance, is around 78%. In other words, this temporal profile indicates that mice accumulate sensory evidence through whisking, and the larger the amount of gathered information, the higher the probability of making a correct choice. All right, so the next thing that we did was to characterize the complexity of these whisker-based discrimination tasks. For each trial, we extracted a matrix that consisted of a value of different whisker features as a function of time, right? For instance, whether there was a contact or not for whisker 1, for whisker 2, for whisker 3. And then we use different classifiers, nonlinear or linear classifiers, to characterize whether in that particular trial, the stimulus was concave or convex, as well as its choice, the site that the animal licked. So we did that with different classifiers, and we got a performance of 90% and 70% performance, percent correct, respectively. And importantly, what we see here is that linear classifiers and nonlinear classifiers, multilayer perceptrons, perform equally well on the task. Or in other words, the whisker-based discrimination task can be understood as a linear integration task across space and time, where space here, I mean the different whiskers. All right, so based on this spatiotemporal pattern of whisking gathered by the animals, we created two new virtual tasks, what we call the easy and the complex task. The easy task was defined as a linear combination across whisker contacts. By construction, the easy task is a linearly separable task with respect to the whisker contacts. Here I'm showing a two-dimensional representation, but it is actually defined in a three-dimensional space spanned by the three whiskers that were more important. On the contrary, we also had this task, the complex task, which was defined as a nonlinear combination across whiskers. In particular, we were using an XOR, which is the most difficult nonlinear task possible. Or in a three-dimensional space, it's no longer called an XOR, it's called a parity. So by taking the same approach that we used to decode object shape and mouse choice, an insightful result was revealed, which is that linear and nonlinear classifiers all had a very similar performance on easy task, while only nonlinear classifiers performed above chance for the complex task. Therefore, these results provided further evidence that the whisker-based discrimination task is a linearly solvable integration task because we found this exact same profile. All right, so what did we do next? We wanted to analyze the neural representations of the sensory cortex and their particular properties. So while animals performed the task, populations of neurons were recorded in their barrel cortex. In order to characterize the properties of these neural representations during this task, we used an encoding approach where we fitted the firing rate of simultaneously recorded populations from all the task variables that we thought could be influencing the activity of the somatosensory cortex. Like for the behavioral case, we used different encoding models with different levels of complexity. In particular, we used a linear model that only allowed for pure and linear mixed selectivity, and a set of nonlinear models that allowed for nonlinear mixed selectivity. So what did we find? We found that the nonlinear encoding model with only one hidden layer was the model that predicted the best the activity of the population activity in the barrel cortex. And it is important to note that I am showing here the R-square on the held out data. I am not showing here the training results, the R-square for the trained set, because it would, well, because I don't have time, but it would basically increase. The more complex the encoding model, the better we would have our R-square. We could also evaluate the contribution of each of the different task variables on the model performance, what we call delta R-square. We saw that the number of contacts made by the whiskers and the angular position of the different whiskers were the two task variables that influenced the most the barrel cortex activity. All right, so even though we found that overall the nonlinear encoding model was better than the linear one, we found that some neurons still depicted linear mixed selectivity. Here I am showing two example neurons with linear mixed selectivity with respect to whisker 1 and whisker 2 and 3. We also, as expected, found neurons with nonlinear mixed selectivity. This first one example shows a sublinear mixing between C1 and C3, while this second example shows an XOR, so to speak, XOR-like tuning curve with respect to C2 and C3. All right, so I don't have a lot of time, but I will try to go fast. So until now, we have seen that the whisker-based discrimination task can be solved linearly by integration of the sensory cues sampled by the mice. But we have also seen that the neural representations of the task variables in the mouse barrel cortex are better explained by nonlinear mixed selectivity, and therefore they are high-dimensional in the neural space. And then we have to ask ourselves, is this particular representation useful on a broader behavioral scheme? Well, to answer that question, I don't have space. Yeah. To answer the question, we used again the easy and complex task that I defined earlier in this presentation and tried to perform them by linearly reading out from the neural activity. Importantly, instead of using the raw neural activity, we use surrogate data created with the different encoding models. What we saw is that the easy task, neural activity created with the complex encoding models degraded performance on this task. On the contrary, for the complex task, only neural activity created with nonlinear encoding models had a performance different from choice. Therefore, even though the whisker-based discrimination task consists of a linear integration tasks on the whisking space, The neural representation of this task in the somatosensory cortex would allow for a complex discrimination with respect to the input variables while maintaining a good generalization properties on a simpler task. All right, so let me recap very quickly. We have seen that mice learned to perform a whisker-based discrimination task, and sensory evidence was accumulated throughout the trial by whisking. We also have seen that the shape discrimination task can be solved by linearly integrating the spatiotemporal pattern of whisking. Populations of neurons in the mouse barrel cortex show nonlinear mixed selectivity with respect to the task variables. Whisker context and angular position are the variables that explain neural activity the most. And finally, the neural representations in barrel cortex allow for complex discrimination while maintaining generalization properties in simpler tasks. Yes, and that would be it. Thank you very much.
Thanks for your talk, Ramon. So let's wait for the questions to show up. But in the meantime, I have a question for you. So your results said that, you know, the number of contacts and the angle of the whisker, like best among the behaviors, best explain the neural activity, right?
Yes.
So have you looked at any other sort of mechanical variables, like say the stiffness, for instance? Because I've seen some work by, say, Mitra Hartman on, you know, how mice modulate like the stiffness of their whiskers and so on. So have you tried to look at like sort of behavioral hidden variables like the stiffness?
Yes, that's a great question, very important. So as I told you, this work builds up upon a previous manuscript that has been published already. Well, it's a preprint, and in that study, I highly recommend you reading it if you are interested in this. We go through all the possible whisker variables like stiffness, curvature. Also, we were also using like task variables like previous reward, current reward, like everything that we could think of. We just put it inside, and we came up with the well, you can see it there, but the variables that I used in this follow-up study were those that showed up to be the most important for behavior in the previous study. But yeah, it's a completely, yeah, it's a very important. Actually, we're a little bit we were surprised when we saw that the whisker curvature didn't really play a role. What really matters for behavior is the number of contacts and the angle of the whiskers, like the kind of whisking that the animal is performing. Yeah.
Cool. That's really interesting.
Yes.
Yeah. Also, could you maybe speak a bit more about, you know, the nonlinear and the linear aspects of the model? Like you said that, you know, linear explained some results and then nonlinear explained the other results. Could you talk more about that?
Right. So we basically used a multi-layer perceptron encoding model, right? So it's the set of task variables. I don't know why I stopped sharing, but let's do it with the hands. So it would be task variables, neural activity, and then in the middle, you can have no hidden layer, right? So this would be the linear model, or in other words, linear regression, right? But you can include a hidden layer with ReLU units, one hidden layer, two hidden layer, or three hidden layer, right? And those, when you include these hidden layers with the ReLU nonlinearity, then you're basically allowing the model to create nonlinear mixed selectivity, well, self nonlinearities, but also nonlinear mixed selectivity between the different task variables, right? And then we feed all the models with cross-validation, and we see that the multi-layer perceptron with only one hidden layer is the one that would explain best the neural activity.
I see. Okay, you have a question. So from Gonzalo, they ask, mice don't touch the surfaces with their whiskers simultaneously. How much temporal integration do you need to solve the task? Is there any mixed selectivity? Is any mixed selectivity good for solving the task?
Okay, so the first part of the question, if I understood correctly, is related to the temporal difference between the contact of the different whiskers. So that's an intuition that, for some reason, like every time we explain this experiment, everybody asks, well, many times we get this question, and we ourselves were expecting that result, that the temporal difference between the whisker contacts was a variable that would explain the best the behavior, but it's not true. It's not about the temporal difference between the whisker contacts. And again, I highly recommend reading this first manuscript we have. but it's the absolute number of contacts that the different whiskers make and the comparison between them. And in the beginning, we were quite surprised because that didn't make any sense to us. If you, as our intuition as humans, I don't know, here's my phone. If I want to recognize this with my hands, basically somehow I will kind of feel, or my intuition is that the difference in time between the different contacts is going to give me a picture, a mental picture of the shape that I'm touching, right? But for we have very stiff and controllable devices here, but whiskers are much more like flexible and unreliable in this sense, right? So that's the explanation that we have at the moment, that it's because they are very unreliable, very flexible. They are not stiff whiskers. The relying on the temporal difference between contacts across whiskers wouldn't make much sense. So it's more about the absolute number of contacts. But again, if you want to check that, read that first manuscript. And then the second part of the question, I forgot. Can you remind me that, Nidhi, please?
Yeah, I think the second part was, is any mixed selectivity good for solving the task?
Right, so the task itself, like we have seen that there's nonlinear mixed selectivity in the barrel cortex, but for the task itself, for the whisker-based discrimination task that I just described, it wouldn't be necessary, because the task is linearly solvable. It can be solved by linearly integrating all these variables. You don't need nonlinear components there. So even though we found that there is nonlinear mixed selectivity in barrel cortex, it wouldn't be necessary. But of course, the animal has been, like the somatosensory cortex has evolved in a naturalistic environment, and this is a very particular task that we are training the animals, but the cortex is optimized for a much more complex scenario in nature.
Got it. So I think we should maybe close the session now, even though there's still a question. Gonzalo says nonlinear mixed selectivity is a very broad term, but we'll have to take this offline now because we need to close the session. Thanks to all the speakers. It's been a great session with really complex real world behavior and neural recordings. So thanks to all of you.